My name is Antti Kause, and I will present the role of selective breeding in the improvement of feed efficiency at rainbow trout farms. And this is part of the AquaIMPACT Horizon 2020 project. Feed conversion ratio has a major impact on the profitability and environmental impact of aquaculture. And this is because feed costs are around 30 to 50% of the total production costs. And feed production itself is a major source of climate change effects. Further, nitrogen and phosphorus of the feed waste cause eutrophication in certain farming locations, and this needs to be avoided. And the aim of the study was first to quantify the changes in FCR and nutrient loading from rainbow trout's farms in Finland from 80s onwards, and secondly, to estimate the degree to which genetic improvement made by a national breeding program has improved the FCR and reduced the nutrient loading. And to quantify the farm performance, we obtained data on commercial farms which have a farming license and are monitored by the authorities, and they are located at the coastal areas of the mainland Finland. And the data ranges from the years 80 to 2016. And each farm provides annual data on feed provided, fish biomass grown, and phosphorus and nitrogen content of the feeds. And using this data, we can calculate for each farm in each year the farm level FCR, which is the total feed used divided by the fish biomass that was grown at the farm. And we can further calculate the specific nitrogen loading to the aquatic environment by having the fish biomass growth multiplied by the phosphorus concentration of the fish minus the total feed use multiplied by the phosphorus concentration of the feed. And the specific nitrogen loading can be calculated in the same way. And on the right-hand side panels, you can see the number of fish farms during this period and also the total fish production. So the production went up and stabilized. Second data comes from the National Breeding Program, and we have a total of 23 year classes starting from the year 92. And this makes 8 generations and 7 episodes of genetic selection. And we have 2 types of traits. Firstly, traits that are routinely recorded in the breeding program. We make routine breeding value evaluation for these traits and we select on them. And we also have traits that have been recorded in separate experiments, but only with 1 or 2 year classes. This figure shows the logic of the data collection and the logic of the study. So we have a freshwater broodstock farm where we make the families. We tag all the families, and each family is split into 2 parts. First part goes to the sea farm, where we record after 1 extra growing season the sea weight, survival, and the visceral percentage. And their SIBs stay at the freshwater broodstock farm, and we record the body weight after 2 growing seasons and 3 growing seasons. All this data is combined, and we make the breeding value evaluations and select the best parents to make the next generation. Now this data is upgraded with the extra traits from separate experiments. So the FCR, fillet percentage, and muscle lipid corrected for the body weight. And we have the genetic parameters, so the genetic variances and covariances of these traits with the routinely recorded traits. And we can combine these genetic covariance structures into a single one. And now we run the breeding value evaluations again, and we are able to obtain the genetic trends for FCR. Now, what is important here is that we don't select for FCR, so the genetic trend that we get is a correlated genetic change because of the other traits that we select for. And this table shows the sample sizes for the traits. And what you can see is that the traits in the routine breeding value evaluation have high sample sizes. This table shows the animal models that we use to estimate the breeding values and the genetic trends. The random effects are the animal, which is related to the pedigree. Then we have the interaction of year and the family tank. The fixed effects are year, the station, sea station where the fish were reared. There are different ones in different years. The gender and the maturity score, the cataract score, and the deformation score. And for the weight 1, which is the tagging weight, we have the cumulative temperature sum. This table shows the genetic parameters that we use in the to estimate the genetic trends and to highlight some key issues. The heritability that we use for FCR is fairly low, 0.07. Growth or body weights correlate genetically favorably with FCR. The FCR has a positive correlations with 2 fat traits, meaning that the high lipid deposition is related to poor FCR. The visceral percentage itself is reversely correlated to fillet percentage. And the fillet percentage has a favorable correlation with FCR. And these estimates come from our previous publications. Now to the results. The farm performance has improved from the 80s around 50%. So to produce 1 kilogram of fish today, we only need half of the feed compared to the 80s. And the phosphorus and nitrogen loading to the sea has at the same time reduced more than 70%. So these are major improvements in the resource efficiency. The breeding program can influence these trends starting from the 93 onwards, and we look at that next. So this is the genetic trend for FCR, and the FCR has genetically improved by 0.19 FCR units. And when we take the farm performance data shown previously, then during this very same period, the FCR at the farms has improved by 0.59 units. So we can calculate that the genetic improvement during this specific time period contributes about more than 30% to the improvement in the FCR. And we can make a simplified assumption that the improvement in FCR due to genetics has a similar impact on the phosphorus and nitrogen loading, assuming everything else remains the same. Now we can also combine these 2 data sets. And in this graph, with green, we have a trend for FCR at the farms, assuming that there would have not been genetic improvement in the FCR. So we have simply subtracted the genetic trend from the trend of the farm performance data. And with the blue, we have the current realized trend for FCR. And now we can calculate that today, due to genetics, the FCR is 18% better at the fish farms. And what we also notice is that without the genetics, there's a flattening curve for FCR. So the improvement would have been flatter without the genetics. Now to conclusions. The results show major improvement in FCR and a major reduction in nutrient loading at commercial rainbow trout farms. And a major part of this improvement is expected to be due to selective breeding, and due to genetics, there's about 18% better FCR at the fish farm. And it's good to note that we make several assumptions here. And for example, the heritability that we use for FCR 0.07 is very low. If that would have been higher, then of course the genetic trend in FCR would have been much, much bigger. And to conclude, this is really a win-win-win. So the same amount of seafood can be produced with significantly reduced raw materials more profitably and with a reduced environmental impact.